This video is looking at the B1 topic cell biology, and it's looking at microscopes. So these are light microscopes because they have light at the bottom of them helping to illuminate what you're seeing on the microscope. Okay, these are relatively cheap but don't have very good magnification. Okay, so they're quite good to have at school. We can see onion cells, we can see cheek cells, we can see kind of big structures, nucleus, cell wall, but not those smaller subcellular structures. Let me go on to the electron microscope, and people get confused with this and they want to call it an electronic microscope because you plug it in. But think to yourself, electron, okay, so if we think chemistry, electrons are really small, okay. These microscopes can see really small subcellular structures. Subcellular means inside the cells, okay. Their magnification is high, their resolution is high, they're really big microscopes and they're really expensive, so that's why we don't have them at school. If you've used a bio viewer, those are photos taken through an electron microscope that we can then view on the bio viewer. So we get the visual of what we'd expect to see under an electron microscope without spending all the money on it. Okay, but remember, it's an electron microscope, not an electronic microscope. Required practical now, we have to be able to write a method of how we prepare and observe cells under a microscope. Okay, so this is a method, this is quite a detailed method. Okay, you'd be thinking about doing this as a 6-mark question, so you might not write in as much detail as this, but we need to talk about the preparation and the use of the microscope. So adding a drop of water to the middle of the slide just helps the specimen that you're going to look at to stick to the slide. You can use tweezers to separate, in this case we're talking about onion skin cells, to separate some of that epidermal tissue, that thin layer between the layers of the onion. That's placed onto the slide, and then we add a drop of iodine because this stains the nucleus so that it's more visible. And then a cover slip can be placed on top, trying to just tap it gently to remove any air bubbles. We don't always use a cover slip because sometimes actually our specimen lays quite flat anyway, but it just helps to lay the specimen flat so that we have a clear focus on the slide when it's under the microscope. We've then prepared our slides, we're up to 0.6 now, and we place it onto the stage of the microscope. Think of like a stage in a drama performance. We put what we want to look at onto the stage. And we start off with the lowest power objective lens. We get a good focus on this so that we can see it, and there's 2 adjustment knobs on the microscope. We've got the coarse adjustment knob, and then we can go to the fine adjustment afterwards. Once we've got it onto the lowest power and we can see it, we can turn the eyepiece lens to the next, sorry, not the eyepiece lens, we can turn the objective lens to the next setting, so we've got a higher power of magnification. We can then focus again, and that might be all we need. Alternatively, we can go to the highest magnification and look at our slide through that, every time using just the fine adjustment knob to get a clear focus on it. We have to use this calculation to work out magnification, size of image or size of real object. So you will need to use your formula triangle to rearrange the equation, and you need to be really careful with units because you might have something measured in millimeters, something measured in micrometers or micrometers, and you need to make sure that you've got that conversion. They must be in the same unit before you use that calculation. The other thing I want to say, just going back to this picture of the light microscope, we've got a lens in the eyepiece at the top there. We've also got the lenses on the objective lenses lower down. To work out the total magnification of the microscope at any point in time, we take the magnification in the eyepiece and we multiply it by the magnification in the objective lens. So let's say, for example, in the eyepiece I'm using the ×10 magnification, and on the objective lens I'm using ×4. So 10 times 4 is 40, so the total magnification will be 40. Let's say I increase the objective lens to 10. 10 times 10 is 100. So I've got 100 times the magnification now. And just to talk about the last thing, standard form. Sometimes with these calculations, numbers are incredibly small. So just to do a small lesson on standard form conversions, okay, it's about how many times we move the decimal place. And we can see on the top example there, it's 6.2 times 10 to the 9. So we've got to move that decimal place 9 times to the right in order to get the real number out of standard form. Similarly, but in reverse, if we have a negative number, so here we have 4 times 10 to the minus 6, we're going to move the decimal place 6 places the other way. Okay, so this time it's going to go to the left, and that gives us a much smaller figure, but you can see how it's actually easier to use it in standard form. Some people like to take the numbers straight out of standard form onto their exam paper, work them through and then put their answer back into standard form. That's the method that I favour. Some people just like to use their calculator, put the information in standard form and just work in standard form throughout. Either way is absolutely fine. Hope you found that useful. Thank you.